Welcome to Mad Biology. My name is Mr. Donnelly. Today we're going to be talking about the non-Mendelian inheritance, also a lot of times called alternatives to Mendelian inheritance. So what does that mean? Well, first of all, we've already talked about in a previous video the actual Mendelian inheritance, which is usually there's a dominant allele and a recessive allele. If the dominant's present, then you see the dominant characteristic. If the recessive is present in a homozygous fashion where there's two recessive alleles, then you'll see the recessive. But if there's a dominant there, it dominates the actual recessive gene. In this case, what we're going to look at is we're going to look at the actual Mendelian alternatives. So mainly that's codominance and incomplete dominance. Codominance and incomplete dominance. How does that work? Okay, so codominance means the two can actually show the dominant characteristic at the same time. The incomplete dominance is when the two actually blend the phenotype. So in other words, they're going to show a blending of the two different characteristics that are dominant and recessive. So let's look at a couple examples. So snapdragons are a specific species of flowering plant, and what they do is when they are crossed, they produce three different colors. Those three different colors are red, pink, and white. So at first this puzzled biologists because it wasn't just two versions of the characteristic. There wasn't a dominant and there wasn't a recessive. There were three. So what was this third one called? What was it? So when you look at it, it's color driven. So you notice that the dominant is red, the recessive is white. So red and white blended together is pink. So what this does is this tells you that it's a blending of the two characteristics. We're going to actually call this incomplete dominance. Okay? So incomplete dominance in this case, we're going to take and we're going to cross two flowers and see what the actual chances are, or the probability is that we could actually get in this case, a variety of the flowers. So we're going to start off by cross-pollinating one with a white flower and one with a pink flower. Okay? Now that I've told you that it's an actual incomplete dominance problem, you can go through and start solving this. So in this case, you're going to have the white flower, which is going to be little r, little r. Then you're going to have the pink flower, which is going to be capital R, lowercase r. So when we take and we cross these two, we can get a big R, little r. Okay, note that we're bringing it in from here and here. Okay. And you can also get a little r, which that is actually going to give us the phenotype of white. So, if we cross a white and a white and pink flower, we're going to get white and pink flowers out. Well, that's not really exciting, but what is exciting is the fact that we have three different color varieties. So let's look at another problem. We'll go down here to number 5. So on number 5, you've got two flowers. They're cross-pollinated. One's going to have a white flower, which is little r. Okay, remember from the last video that the lowercase r is actually going to be the recessive gene, and we use the first letter of the dominant gene, which is capital R and lowercase r for the actual allele abbreviations. So we've got two flowers. We've got one that has white and the other has red. So we cross it. We've got red and white. And what you're going to end up with is all pink flowers. So if you remember from the last video, that would mean we have a 100% chance, okay, or 1.0 probability of getting pink flowers. Okay, cool. So that's incomplete dominance. So now let's go over here and let's look at another example. In this case, we're going to look at cattle, which in cattle we see codominance. Well, codominance is not incomplete dominance. So codominance means that both traits are dominant and they can show both at the same time. Now, there's a little bit of debate here. Now, why do I bring that up? Okay, so at the end of this worksheet, I took and I posted some websites. Now, specifically, we're going to have it right here, which is going to be the changes of codominance in color and cattle. Why do I do this? Not to confuse you. Okay, I want to give you the actual opportunity to understand further what's really going on. So remember in my last video I talked about Reginald Punnett, and I talked about the actual studies that have been done over time where we really didn't understand genetics. Well, now we kind of do understand it a little better, and things aren't as simple as a Punnett square makes them seem. So with that being said, we're going to keep it simple. So in this case, what we're going to do is we're going to take and we are going to cross two cows, and one is going to have red fur and the other is going to have white fur. What are the possible outcomes? Well, there are two ways of doing this. Some teachers will take and they will use capital letters, R for red and W for white, and they use both capital because they're both dominant. Some other teachers will also take and they will come in and say C for color and use an exponent, R for red and W for white. In this case, I like to stick with the simpler version, mainly because, well, you're not writing real small, and it's easier to make mistakes when you write real small and try to read your writing later. So in this case, what we're going to do is we're going to cross a red and white furred cows. So you have RR, and you're going to have capital W, capital W. So when you take and you do this cross, it doesn't really matter which letter goes in front. But you notice that you end up with RW cows, and it's 100% probability, or excuse me, 100% chance, 1.0 probability. Okay? So what does this look like? Well, this is what we call a roan cow. Feel free to look it up. I didn't want to pull pictures in that might have some kind of legal problems. So in this case, a roan cow is going to have red and white patches. So you get these patches. One of them is going to be red, and outside is going to be white. And you start to see that there's this patchiness. Well, part of that's due to development. Another part of that is due to this actual gene. And that's where I say it's a little more complex than what one Punnett square can lead to. But for the purposes of a high school biology class, this is what you need to know, okay? Another common example of codominance is the human blood type. So here we're going to look at ABO blood type. We're not going to look at the RH. The RH is the plus and minus that you may hear about, like O positive, O negative. We're only going to look at the O part of that. So we're going to have two children. They're going to have a child. The mom is heterozygous for type A blood. You remember that exponent I talked about up here? We're going to use that now. We have to. Okay? So in this case, we're going to take and we are going to use I. I is your dominant letter, little I is your recessive letter. Why are we using this? Because we have three different possible outcomes. We have A, B, and O. Okay? So that's where the ABO comes from. What are the possible outcomes that the child could have if the mom is going to be heterozygous for type A? So that means she's going to have a dominant A allele there. And it doesn't say she's AB, so that means that this second allele has to be a lowercase I, which is the allele that we use for O blood type. The dad is going to be heterozygous for B blood type. So that means that dad is going to have capital B, and he's also going to have that little I. What are the possible outcomes that they could have? They could have a child. This is where I'm saying it gets kind of hard because you're writing small. A child with AB, and they could have a child with B. They could also have a child with A blood type, and they could have a child with O blood type. So you can look at this and you can start to see where this might be kind of cool to use. You could predict someone's child's blood type. Okay, well, where would I want to use that? Well, for a while it was used to actually figure out if two people were the parents of the child. So they went through and they actually started to figure out what the blood type of the mom and the dad was and see if it was even a possibility for the child to have that blood type. But they found that this is not the most reliable. It's just 1 point on the actual genetic makeup of a human being. So that variety didn't really help out too much. All right. So I'm going to leave the rest of these for you. You've done previously this genotype-phenotype probability chart that I've put on my previous worksheet, so I'm asking you to do it now on this also. Okay, now we're going to move on to the second worksheet that I have. This one is labeled as Mendelian Alternatives or NonMendelian Inheritance on my website. So now you've decided that you want to actually go and become a botanist. You remember a really cool example that this high school biology teacher used to talk about with snapdragons. So you go down to the local nursery, you find a pink snapdragon. Okay, so now you know you're dealing with capital R, lowercase r. Okay, and you decide to add it to your collection of snapdragons because evidently you've become a snapdragon collector. The next year you notice that it comes back with 4 stalks. 1 is red, 2 are pink, and 1 is white. So using your knowledge in the tables provided, please explain why your snapdragon is not mythical. Okay, this is a flipped problem. So to tackle this, we kind of have to do a little thinking. So you know that you've got 4 stalks possible. You only ended up having 1 pink plant. Okay, so you have 1 pink plant that was this capital R, lowercase r. And with your 4 stalks, you had 1 is red. You had 1 as being white. Actually 2 that are pink. So this ratio kind of tells me something about it. It's a 3-to-1 ratio or a 1-to-1, which is something that's very common. You can see this when you take and look at a typical 4-square Punnett square. So where do you see it though? You see it when you cross 2 heterozygous individuals. Okay, so what was our original parent? Our original parent was a heterozygote. So if I take and I look at it, I've got 2 heterozygotes that I'm crossing. Oh wait, I get 1 red, I get 2 pinks, and then I get 1 white. Oh, doesn't that represent what I previously had? It does. Okay, so it's not mythical. I actually, by just looking at this and looking at the numbers, I can figure this out. Well, let's go ahead and fill out the table here. In this case, the genotype itself, we've got genotype, capital R, capital R, capital R, lowercase r, lowercase r, lowercase r. Now our phenotypes, we have red, we have pink, and we have white. Okay. What are the probabilities? Well, we can base the probabilities based on the number of individuals that we had in our population. Okay. So the probability in this case, we've got a probability of 0.25. And we have a probability here of 0. I'm going to put a 0 after it, 50, and 0.25 again. Okay, so how do we explain our results? Well, based on our probability, we have actually determined that we should have in our Punnett square 1 square dedicated to the red, 1 to the pink, 2 to the pink, and 1 to the white. And so we go back and we look at what we had originally hypothesized over here, and we see that that is the case. Just because of our familiarity with, or how familiar we are with the actual data itself and how Mendelian or non-Mendelian characteristics work. Okay, cool. So on here you've got 2 other problems. Now you decide to be a cattle breeder, and then on the other one you're going to look at being a blood scientist. If you have any questions, please feel free to leave them in the comment section below. This is Mr. Donnelly with Mad Biology. Please feel free to like, share, subscribe, and share with your friends. Thank you.